And our next speaker is Dr. Tyler Dreaden, and he will be talking about the current state of Laurel Wilt science, including some of that kind of exciting work that Bud mentioned. So Dr. Dreaden received a Bachelor of Science in Forest Resource and Conservation in 2006, a Master's and PhD in Forest Pathology in 2010, 2014 from the University of Florida. He has conducted Laurel Wilt research as a graduate student, postdoctoral scholar, and in his current position. So a real world expert on Laurel Wilt disease. He joined the USDA Forest Service Southern Research Station as a research plant pathologist in 2015 and is part of the Forest Health Research and Education Center on the University of Kentucky's Lexington campus. So happy to have you, in Lexington and on the UK campus, and happy to have you joining us today.
Thank you for the introduction and the opportunity on me. Okay, so I'm going to give a little bit of an overview of some more recent findings. But this is, so I will not be presenting much of my own research. So this is research by a whole bunch of people. And it's just going to be a very brief overview of the studies. But I do provide the citations for everything if you'd like to, pull up the papers and look into the studies in more detail. So as Bud mentioned, Laurel Wilt disease is a little complex because it's caused by, the fungal pathogen Rafflesia loricola, which is a nutritional symbiont of the red bay ambrosia beetle, Xyloborus fiberatus, and it affects members of the Lauraceae family. So all of these things have to be involved to get disease development. So we'll talk about each of these in a little bit of sections, but we'll start with some recent studies on the pathogen. So you can get rapid symptom development. So Hughes et al. Inoculated small red bay trees, so about 1.5 meters high, with a range of spore concentrations. And he was able to get disease development with as little as 100 spores inoculated into the trees. But with over 1,000 spores, so here in panel B, we've got consistent symptom development, and with a few more spores, you can see disease developed very rapidly. And then one of the studies that Bud mentioned and he conducted, it was in larger trees, so 17 to 39 centimeter DBH trees. He got symptoms were seen after 6 weeks in 3 of 10 trees. And after 12.5 weeks, symptoms were in 9 out of 10 trees. So even in large trees, you can get symptoms develop fairly rapidly. The pathogen isn't in the xylem very abundantly. So this study was conducted by Campbell et al. So she took the pathogen Rafflesia loricola and inserted a green fluorescent protein in it. And what that does under fluorescent microscopy, it'll glow bright green. So if you look at the figure at the upper left, this arrow here, so this green is the fungus growing, and this red background is the host stem. So she inoculated trees with this green fluorescent protein labeled pathogen, so she could try and see where the fungus was in the xylem and how abundant it was. And what she found was immediately after sampling and then mounting the slides and looking, she could not see the fungus at all, just couldn't do it. And after she put these sections on slides with growth medium, then after 3 days, she could just start to see the fungus come out of some of the xylem and cells. So the image on the left is after they incubated the slides for 3 days, but then after 5 days of letting the fungus grow out, then they could clearly see the fungus. So it was in the wood tissue at a very low amount. So she also quantified the percent of the xylem lumina that were colonized by the pathogen, and it was very low. So it was less than 1% of the lumina were colonized. So the pathogen is causing symptoms to develop very rapidly, but yet it's not overly abundant in the xylem tissue. So this study is by Inc. et al. So similarly, they inoculated host trees to try and see, well, if the fungus isn't growing in all the xylem and physically blocking them, why are these symptoms developing so rapidly? So they used SEM to image the trees. So this top panel. So this is a mock inoculation where they didn't inoculate the tree with the pathogen, but did the same wounding procedure. And then in B through C and F, this is where they inoculated the hosts with the pathogen. And what they found was abundant tyloses production. So these arrows are tyloses developing in the vessels and blocking movement of water in these vessels. And you can see by 48 days after inoculation, almost all the vessels are blocked. So again, they tried to find the fungus in these hosts. So they stained it with they stained the fungi, and you can see the fungi are glowing bright green, and they're labeled with the red arrows. But just like with the Alina study, they had a hard time finding the pathogen in the host, but yet the hosts seem to be producing the tyloses, blocking the xylem and causing the wilt. And it's not the fungus physically growing and blocking all the vessels or causing symptoms to develop. So it leads to the question of, what are the hosts reacting to? So Shin and Smith at University of Florida, they investigated inoculating trees with sterile culture extracts, but did not get symptoms to develop. Calero and Zhang compared genomes of Raphiolarcula with related pathogens. So that's these two bottom figures, trying to find genes that were shared between pathogens, but absent in non-pathogens. And they found some candidates, that were genes of interest, but it was no smoking gun. So they have some genes they like to pursue more that might be involved in the host reaction to the pathogen, which is producing the tyloses, but they haven't narrowed it down yet. So more work to be done. So as Bud mentioned, this is Mark Hughes and Jason Smith's work. So this is part of Mark Hughes's PhD work, trying to identify resistance in red bay. So what he did was he went to stands that were just decimated by Laurel Wilt disease. So in this upper left photo, all of these are, dead red bay. So disease pressure was really high at these sites. And he surveyed and tried to find, single large live red bay. So this one red tree in the center is a live red bay, and it's surrounded by dead red bay. So he selected these as candidates to screen for resistance. So he then propagated them, clonally grew them up, and then planted them in a field for a resistance screening trial. So again, this is Mark Hughes' work, and he provided this slide. So went to the field, found candidates for screening for resistance, propagated them, and grew them up in the field, and then inoculated these trees by, drilling a small hole in the base and pipetting in a spore suspension, and then waiting for symptoms to develop. So this is his work in 2014. So most of the trees were like the one on the upper right, and completely wilted after 12 weeks. But there were a handful of clones, like the one on the left, where only a portion of the crown or a branch would wilt, and the rest of the tree would remain healthy. So he's calling these tolerant. And the ones that survived the 2014 trial, he did go back the next year and inoculate them again, and he got a similar result where a portion of the crown would wilt, but the tree itself would not die. So it's pretty exciting and promising that they found some host resistance or tolerance. So next steps for that are to try and identify more tolerant individuals. And then once you have a number of these tolerant individuals identified, you can start breeding the tolerant clones to try and increase the resistance. And then try and deploy a restoration strategy and how we hope to deploy these tolerant individuals. But it's also, and we'll get into it more in a minute, but we want to limit future introductions of the pathogen or vector. As Bud said, there's a single introduction, and if new genotypes of the pathogen are introduced, it's possible that they could overcome the tolerance that Mark has identified. So now we're going to shift gears a little bit and talk about the red bay ambrosia beetle, Xyloborus glabridis. So as Bud said, the beetles are attracted to host volatiles and larger diameter stems. But interestingly, host attractiveness varies with the stage of symptom development. So in this study, figure 1 at the top right, so they have control trees and then trees that they inoculated. And before the inoculation, both the controls and the ones they were going to inoculate in the future are equally as attracted to the beetle. But 3 days after inoculations, the infected trees are less attractive than the uninoculated trees. But then by 10 days after inoculation, this trend reverses, and the infected trees are more attractive to the beetle than the control trees. So this led them to investigate what volatiles are given off at different stages of infection. And they identified what chemicals were attractants and which ones were repelling the red bay ambrosia beetle. And then they did a study where they put these repellents, so verbenone, a combination of verbenone and methylphenolate in combination on hosts to try and see if they could repel the red bay ambrosia beetle. And what they found was verbenone and the combination. So this is the number of Salmonborus glabratus hitting a trap per week. And this is weeks of deployment. The controls had a large number of red bay ambrosia beetles, but the best repellents were just as many hits as non-hosts Laurel Wilt disease. So this led them and others to wonder if a push-pull strategy might be an option. So I snagged this figure. So a push-pull strategy is where you put a repellent on your crop to try and push your vector away from your crop, and you put an attractant in a trap to try and pull your vector to the trap. So this is hypothesized, might be part of a IPM, and it is starting to be studied, but more research is needed. As Bud said, it only takes one female to start a new infection in a tree and start a new colony. So it's going to be hard. So as Bud said, the nutritional symbiont of the red bay ambrosia beetle is Rafflesia loricola, and it's transported in the Mycangia beetle. But the Mycangia also contains multiple other fungi, and the community complexity, or the number of different taxa found in the Mycangia, and the actual taxa in the Mycangia varied geographically. So here's an example. You can see the Mycangia membership, in Miami-Dade County in Florida was different than Alachua and Highlands County. And it varies via location. But we did take these fungi that were found in Mycangia, including the known pathogen Rafflesia loricola, and inoculated host, to see if any of these other fungi might be involved with symptom development, but only the known pathogen caused external symptoms to develop. So now we'll shift a little bit and talk about the nature and structure of the Laurel Wilt introduction. So as Bud said, the initial detection was here near Savannah in 2002, and it made several jumps in the distribution. So a few of these, as Bud mentioned. So this led to several questions. How many introductions in the U.S. has there been from Asia? And are these new populations, disjunct populations? Are they the result of moving the same genotypes around that are already in the U.S., or are these new genotypes coming in from Asia? Also, what is the source of the U.S. population? So Hughes et al. Studied the COI gene, and they found only one genotype of the red bay ambrosia beetle in the U.S. And then two population genetic studies looked at the fungal pathogen and its diversity in the U.S. and Asia. And they found limited diversity in the U.S., but more diverse populations in Asia. And both of these support this single US introduction hypothesis. So a little bit more on the population genetics of the Laurel Wilt pathogen, Rafflesia lyrica. So the figure on the right is a minimum spanning network using microsatellite markers. And it represents populations from the US. The multi-locus genotypes from the US are the purple nodes. Taiwan is the turquoise, Japan green, and Burma is in red. And the numbers represent each individual isolate of the fungus. Isolate numbers in black are mating type 2, or have the MAT2 mating type, and isolate numbers in blue represent mating type MAT1. So in the US, we found very little diversity. So the majority of the isolates were this multi-locus genotype 5. But there was also 2 minor multi-locus genotypes found in the US. And there was only a single change between each of these genotypes. So they were very closely related. And we only found 1 mating type in the US, whereas multiple mating types were found in Asia. And one of the most interesting findings was the main genotype that is present in the US. That same genotype was found in Taiwan, which begs the question if Taiwan was the source of the US introduction. But very little of the beetle's native range was sampled, so we can't draw any firm conclusions. But we did find that isolates from Myanmar were quite different than the isolates found in the US, Taiwan, and Japan. This is also highlighted in the DAPC plot here, where the isolates from Myanmar were quite different from the isolates from the US and Taiwan. So this supports the single introduction hypothesis for the US. So there's estimated to be over 300 million red bay trees killed in the US. And this is based on 2014-2015 data. And where the mortality is, it's a quite high percentage of hosts, so 70% host mortality in these areas, and over 300 trees per hectare. But as of now, there's no estimates for the number of sassafras killed. So they estimated the rate of spread, but this was based on red bay and sassafras host density, but it spread much faster than estimated because of the host people moving infested material around. For example, they estimated it to get to Texas in 2040, 2035, but it was there in 2016. So it's moved much faster than we had hoped due to people moving infested material. But this work was done using red bay and sassafras host density, and didn't model spread in sassafras. And we don't have a good idea of how fast the disease will be able to spread in sassafras. So early on, there was hope that cold temperatures in winter would kill the vector. And some work by Formby et al. Um, investigated this, but unfortunately, you can see in the light gray area here, winter mortality is not likely to help limit the vector, at least for most of Kentucky. And this model, this work was, based on some lab studies, but since we're seeing the disease seemingly spread in Kentucky pretty good, it must be fairly accurate. And with that, I just wanted to share some more resources. So here are 4 good review articles, and the bottom link is for the county distribution map, and they keep that updated. And with that, I would just like to say thank you for letting me talk today.
Oh, wonderful. Thank you for talking with us today. Really appreciate it. And I see that we had a few questions in the chat that maybe we can visit briefly before we move on to our next speaker. But just as we wrap up this first kind of morning segment, a big round of virtual applause for you, Tyler, and for Bud as well. So one of the questions that we received in the chat that I think has been touched on, or this was in the Q&A, was what the mortality rate for sassafras infested with Laurel Wilt disease. Is there any natural or signs of natural resistance in what we've been seeing in Kentucky? And so I know that Bud and I chimed in on that over the chat. Bud, do you want to, or Tyler, would you like to kind of share your thoughts and observations on that?
So like we mentioned in Mark's study, where he went to decimated stands and found a few surviving redbay, we've started a study like that for sassafras. And I think we might talk about that more at the end. So we have hope, but we haven't found or tested any yet. So we will see.
Yeah, we've been working in a very large property, Tyler and I, looking for sassafras that are still alive. And I think we're now up to, I don't know, of the many, probably hundreds of thousands of trees that were there before, 4, 5, something like that, that are still alive. Now they show symptoms of Laurel Wilt disease, but maybe they have more resistance. Hopefully, maybe they're just lucky and only time will tell. Bud, you were going to say something.
Yeah, let me see if I can get my video on here. Yeah, we've seen very high mortality rates in sassafras as well, not only on the coastal plain, but also in East Tennessee and Central Tennessee and Kentucky. A year or two ago, I guess it's been 2 years now, right before COVID, we went down to the coastal plain in Georgia as part of Tyler's project, looking for surviving sassafras in areas that Scott Cameron initially surveyed as sassafras stands. And we were able to find, the good news was there were still sassafras in those areas that had had Laurel Wilt for 10 plus years. Almost all of it is at very small diameter. And I mean, I think the biggest tree that we saw is, maybe like that you could just kind of start to get your hand around. It just kind of remains to be seen whether those are somehow trees that escaped. Maybe they were at very small diameter when the beetle population was high, and so they got passed over, and it did not spread into their root system. And so now there's a sassafras cluster there. Is it going to be susceptible to remaining beetles that are still in the area? Kind of, we just don't know yet, but it's an excellent question, and why Tyler's got his project going for that very thing.
And I guess we could branch over into spice bush with that same thing, and that we really don't know what's going to happen with spice bush. Spice bush, for those of you who aren't familiar, it's a smaller, shrub, small tree, and it can grow quite densely in the understory sometimes, but it's going to be much smaller diameter than, your larger sassafras. However, we have seen, spice bush dying that has tested positive for Laurel Wilt disease with extensive beetle tunneling in it as well. So clearly, even those smaller diameters could be attractive to the beetles under the right situations. But where I've seen that, was also a very high level of large sassafras prior to that. So I don't know. Any other thoughts on what this spells for spice bush from you all?
I'll say that several years ago, we did a series of hanging bolt studies. So we cut up firewood-sized pieces of different host material and hung them in kind of a randomized block design in forests that had red bay ambrosia beetles. And we tested spice bush and swamp bay and red bay sassafras and some non-hosts as well. Spice bush was one of those species that did not have hardly any red bay ambrosia beetle hits on it at all, which suggested to us that, well, it must not be very attractive. And we kind of clumped it together so that it had a large silhouette, similar to the other bolts. It just didn't get attacked very much. But that was also when there were other host species available for beetles to go to. And so if you have a scenario where the only laurel species in the stand are red bay or are spice bush and sassafras, and all the sassafras is dead, I can imagine a scenario where then spice bush becomes, it's the only game in town. It's going to get very heavily attacked. Or maybe there's something different about a live spice bush plant compared to a chunk of a spice bush hanging on a rope. And so it's a puzzle, and I think Alexandra probably has as much experience looking at this in the field as anybody. When we first set up a monitoring plot at Jeffers Bend, we really didn't see the disease in spice bush there, only in sassafras. But now, if I understand correctly, Alexandra, it is in spice bush at Jeffers Bend now. Is that right?
It hasn't been confirmed yet, Bud, but yes, near one of our panel traps that's hanging in the sassafras there, the spice bush showed first symptoms of wilt and got a pretty darn good clean sample out of it. So interestingly enough, didn't really notice any beetle hits, but maybe lingering in the soil or something of that nature. So,
and those beetles that you sent me from the spice bush sample in the fort at Fort Campbell were immature adults. They were callow adults from spice bush that you reared out, which suggests that the beetles were reproducing in that spice bush stem, not merely just attacking it and exiting, but those were beetles that were born in that tree. So they do seem to be able to reproduce in spice bush.
Yeah, so some more doom and gloom to not look forward to, but
right.
Yeah, we'll keep a close eye on that Jeffers Bend location. But as you know, it's very heavily populated. The understory is mainly spice bush, so it's looking kind of dismal.
We did have two additional questions that I wanted to touch on before we move to our next presentation. First, are the beetles finding new host species to feed on? It's my understanding, no, they're not moving to other species. Have you all encountered any of that or other things outside of that Lorraceae? No, but there are other beetles that can move the fungus around, which is another concern that perhaps we could talk about. This is not the only ambrosia beetle in town, not even the only invasive ambrosia beetle around. So you'll have found other insects carrying that fungus, correct? All right, I'm seeing heads shake yes, unfortunately.
Yeah, a lot of that work was done in Florida in the avocado orchards,
Ooh,
where
more to
red
come
bamboo
soon.
beetle is very rarely ever trapped. And so the fungus was still moving around in those groves, probably underground, but also by other vectors, like some of our native and naturalized non-native ambrosial beetles. So Romina may have more information on those as well.
Oh,
Okay.
good. More to come on that soon. Excellent. All right. Well, then one more question. Can the beetle be spread in mulch even though it was chipped up?
I would say that it's possible, but maybe improbable. James Johnson, I remember James, who was the forest health specialist in Georgia for many years, felt very strongly that Laurel wilt spread in coastal Georgia along a railroad line that was moving hardwood chips to a pulp mill. And when you see pictures of this beetle, or if you've seen it yourself in your hand, it doesn't take very much imagination to realize that beetle could survive in a chip. Even, something that just came through a chipper and chipped everything down to square inch size pieces, a beetle could survive in that chip. Is chipping it up going to reduce the number of beetles? Absolutely. The wood's going to dry out. You're going to chop some of the beetles up, but it's not going to be 100% kill, especially on heavily infested materials. So it is possible.
But I guess chipping, they will no longer be able to utilize that tree as a resource to reproduce on going forward. So
that's right.
All right, well, thank you very much. If you have other questions, you can put them in the Q&A. And again, a huge thank you to you, Tyler and Bud. Obviously this is just a really complex problem, and you can't help but think of something like Dutch elm disease when you think of Laurel wilt. The vector side of things, the fungal pathogen side of things, the tree defense side of things. And I think it's just really exciting having you all and other great scientists working on this issue. Hopefully 50 years down the road, we'll be somewhere better, with Laurel wilt disease going forward, because we have all of that. So much appreciation to each of you.